I'd now like to introduce Mafalda, who comes from CRG in Barcelona. For those who work at Gen, they probably know it well. Mafalda's background, a lot of Bayesian learning and generative models. Particularly predicts the effects of genome variation on phenotype. So obviously this has got direct impact on the diagnostic yield of patient sequencing. Mafalda's talk today is on, I can read it from here too, models with Jesus, my English is terrible. Models with interpretable scores for discovering pathogenic variants.
Well, thank you very much.
Over to you.
Thank you very much for having me here. I'm super happy to talk about my work. Yeah, so I think we're going to dive a little bit more into sort of the research enabled by the kind of infrastructures that Tim was talking about. And what I would like to talk to you about today is an unmet need in clinical genetics of trying to pinpoint pathogenic variants in patients. So as sequencing is becoming available for diagnostic care and preventative care, we have this necessity of identifying what could be the causal pathogenic variants in a patient. And today I'm going to be talking precisely about identifying pathogenic missense mutations, and this is really a needle in a haystack kind of problem, because as an average person has about 10,000 missense variants in the genome, to try to identify what could be the pathogenic variants is kind of like a needle in a haystack problem. There's been a lot of development over the last decades in statistical genetics to try to collect bigger and bigger cohorts of patients and across the population, and develop tools in GWAS and protein analysis, but these methods still remain limited, especially in the long tail of rare variation. Often there's sort of gene-level statements rather than variant-level statements. And this is a problem for pathogenic and oligogenic disease, but it's also a problem for monogenic Mendelian disorders, where you would have imagined that it's where it's easiest to identify what is the causal variant, because it's kind of like a large effect variant that is driving the disease. But actually, the vast majority of patients that suffer from Mendelian rare diseases still don't have any kind of genetic diagnostics. And part of the problem is that being rare, these diseases are just harder to study. It's harder to put the cohorts together, and it's also, even when we have large cohorts, they're limited in scope because it's quite hard to identify what is driving diseases that are as rare as one. So one approach that has been very fruitful in the last decade is to kind of decrease drastically this haystack by just looking at de novo variants, so sequence trios, and by that I just mean sequence the parents of the patient to try to identify the de novo variants. And this is powerful because it has been shown that severe disorders, rare disorders, are often driven by de novo variants. And this is kind of here in this plot, you just have an example of a cohort that was actually put together by people here at Sanger from the Matt Health Group of 31,000 trios of patients with developmental disorders. And you can see that there is a large excess of de novo missense variants in this cohort, kind of like pointing towards the idea that these patients have causal missense de novo variants. But the reality is that even if this looking at de novo variants kind of reduces the problem and make it easier, most of these excess missense variants are still unexplainable and outside of genes that we know are involved in developmental disorders. And also trio sequencing is also difficult to put together. It's expensive and it involves the participation of both parents, which in many cases, in many settings, it's quite hard to obtain. But the flip side of this is that it points towards quite a simple genetic architecture of these disorders, and really is just saying that if we just are able to find that causal variant, we might be able to diagnose a lot of patients. So this seems like a great setting for computational biology and for deep learning to try to address. So the idea is that we have what we would need, I guess, to develop a model that is able to identify or pinpoint causal mutations, pathogenic variants in diseases as rare as one, and perhaps even across the whole exome rather than just looking at de novo variants. We just need a model that has a meaningful notion of severity, kind of an interpretable notion of severity, such that I can identify that there are some variants that are pathogenic or might be involved in some disorders, but there are some that are really outliers and might be driving extremely pathogenic and extremely severe disease. And for this, we need tools that satisfy two conditions. First of all is that, like I was just saying, this pathogenicity score really meaningfully reflects variant severity. And also we need this score to kind of hold for the whole exome, because we want to study variants across the whole exome of these patients. And I'm going to try to make these more concrete. So some years ago, together with collaborators, we were exploring the idea of looking at variation across diverse species as a way to identify pathogenicity in variants in humans. And the idea is super simple. So if we just look here, it's protein sequences, or it's supposed to depict protein sequences across a protein family, across diverse organisms. The idea is if we have a model that is sufficiently expressive, that can capture patterns that are being preserved across millions of years of evolution, preserved, co-evolving together, then we are able to capture constraints in sequence space that might be related really to function of this protein family. And the idea is that once we have such a model that describes the distribution of these sequences, we can then ask if a new sequence with a variant that maybe we never saw before in a human, how likely it is according to this model. And the idea is that if this sequence is an outlier according to this model, maybe this variant is disrupting some important pattern of constraint in sequence space, and it's probably associated with lack of fitness in this protein and probably associated with disease. So this is kind of like a super simple idea, and but actually it works pretty well. So for this specific work, we used a variational autoencoder, but there's different tools now that use different kind of deep learning models with kind of same idea and ethos in mind. And so just to give you an idea of the kind of performance of these models, so we call this model EVE, and this is an example for P53 and P10, and here magenta are clean VAR known pathogenic variants in these genes, and the blue or teal are known benign variants in these genes, and you can see that the model quite separates very nicely benign from pathogenic variants. And not just that, but it has similar performance to deep mutational scan experiments, so experimental assays to try to address this exact same question. And we did this at scale to every single gene that we knew was involved in disease. And okay, for some genes we have more data for validation than others, but across the board we see that there's quite good, or these models have quite good power at separating benign from pathogenic variants. And across the last few years, there's been a lot of development in this kind of space. There's a lot of tools that kind of address this question, and they are actually quite excellent at separating benign from pathogenic and predicting clinical data like I was just showing you. Now the problem is that the interpretation of these models is not very clear. And let me just give you an example. When we look at the distribution of scores for genes that are haploinsufficient, so definitely involved in severe disorder or involved in dominant disorder, versus loss of function tolerant genes that are effectively non-essential, what we see is these models have very similar scores for both of these kind of families of genes, where you would expect that you would see a much stronger tail of pathogenic variants in genes that are involved in disease compared to genes that are non-essential. In the end, what is happening is that these models are excellent at ranking variants within genes, but they really fail. So they're very good at predicting if a variant is going to disrupt molecular function in a particular protein family, but they fail to capture how impactful that gene is for human disease, and therefore how this relative severity across genes really impacts human health. Another way to look at this is that they kind of, all of these models provide a continuous score, but the interpretation of this score drastically changes from model to model. Instead of having a score that maps to disease severity, what most models are providing is a score that actually reflects the probability of a variant being labeled as pathogenic or benign. So many models have a bimodal distribution. Just here is an example of alpha missense, where close to 0 are all the benign variants and close to 1 are all the pathogenic variants. And instead of this score being reflective of severity, it just basically says the model believes that this variant might be involved in some disease at some point. So with this kind of model, it's really impossible to identify severe pathogenic variants, as you would have, all the variants mapped to a score of, I believe this is a pathogenic variant. In a sense, you can already see that this distribution of scores cannot really be a distribution of severity, because it's bimodal, which goes completely against your expectation of negative selection on severe pathogenic variants. So to try to gain a notion of interpretation in these kind of models, we opted to take a strategy, which I believe is a strategy that we're going to hear over in this conference, which is to build a simple model, a model where we can have explicit model assumptions on top of a highly expressive model, that's a deep learning model that really is not very explainable and not very interpretable. And to do this, in this particular setting, we leverage variation on 2 different timescales, so variation across the tree of life, like I just described, that just models constraints across evolution and constraints in protein families and protein molecular function, and also model the variation across a human population, so model human-specific constraint that hopefully maps to human health. The way we do this is to first compute these variational anticoders, large language models across diverse organisms for every single protein in the human proteome. So we have about 20,000 of these models, and for this we get an evolutionary score like the one I just described that has very good variant level resolution at distinguishing variants that are impacting protein function and molecular function, but are not very good at distinguishing if these variants are important for human health. And then on top we change the problem and rephrase it as the capacity for the model to predict if a variant is seen or not in the UK Biobank. So we just use this as a very simple model. Well, it's just like a Bernoulli process with a Gaussian process. In fact, if that function f was just a linear function, this would just be logistic regression, but we allowed it to deviate a little bit from linearity. I'm happy to talk about this later if you're interested. But the key aspect here is that by doing this, because we have variation across the whole population, across the whole proteome, by predicting if a variant is seen or not in the UK Biobank allows us to have some sort of information sharing mechanism across all of these protein family independent evolutionary scores. And what we get is that this model rescales the scores for each protein family according to how important it is for human health. The result then is, bringing back the example I showed you earlier, is that we see that for haploinsufficient genes, now we really see here, we call this model POPI just because it has population data. We can see that more negative values are more pathogenic, and we see here a tail of negative variants in genes that are involved in disease compared to genes that are effectively non-essential. So the kind of behavior that we would expect from this kind of model. And when we look at the distribution, so this is the distribution of scores across all the variants in the UK Biobank, in opposition to this kind of bimodal distribution from models that are just trying to predict if a variant is benign or pathogenic within a protein. What we have now is kind of a bell-shaped curve where you can see that it is negative tail corresponds to the more pathogenic variants across the population. A little bit more like we would expect according to negative selection on these variants. So what can we do when we have a model like this and we apply it to try to identify pathogenic variants in disease cohorts? So we looked at, again, this 31,000 trio cohort put together by the group of Matt Hurls, And we see that the model is quite good at distinguishing cases from controls. So these are the de novo variants at the de novo variant level. And what we see is that at the benign end of the score, so around -4, -3, we cannot distinguish if the variants belong to a patient or a control. But what is very interesting is that for scores that are below here around -5, we have, we have about 99% confidence or recall that these patients, these de novo variants are really variants in a patient rather than a control. And you can see that this corresponds to almost like a 10-fold enrichment in variants that are deleterious at these scores. So we can use this to try to identify variants that are potentially causal. And when we do this and see in which genes they are, we recover a lot of the genes that have been found in this study before. We also find a lot of genes that have never been identified in this cohort but are known to be involved in developmental disorders. And we also find evidence for genes that were not known to be involved in developmental disorders before. And actually, since we put this paper in the bioarchives, 20 of these 120 genes have since been identified in familial studies, have been identified being involved in the developmental disorders. So really sort of giving us some confidence and maybe the kind of evidence that we are bringing is meaningful. And I was talking about showing you results of how to identify, pinpoint de novo variants that are causal, but then we can try to do this exercise in trying to identify or pinpoint variants that are causal across a whole exome. So it's a much harder problem. But we again see that in the lower end, more negative end of the scores, we can start to identify patients and variants that are associated in patients. And in fact, we find that in 98% of the cases that we identified with de novo analysis, just by looking at the exome, we recover them and we identify the variants, the de novo variants, as being the most severely pathogenic across all of the variants, included the inherited variants. So giving us kind of a feeling that we can use these kind of tools without trios and really just directly at the exomes of the patients. And this kind of gives the same kind of message, this plot on the right. This is just the percentage of the cases that are putative diagnoses, so people that have variants in genes that are known to be involved in developmental disorders, and with a de novo variant there. And just by looking at their whole exome, we can see that we recover a lot of these, a lot of the variants and causal variants without the need for trio data. So just to summarize, until now, methods did not provide, or methods, variant effect prediction methods, did not really provide an interpretable severity score, and instead provide a score with a difficult to parse, a continuous score that is difficult to parse and with a difficult interpretation and mapping to severity in humans. And by leveraging variation on different time scales across the whole tree of life and across a human population, we managed to build a model that sort of places variants in a proteome-wide spectrum of severity. And this can help us to identify variants in disorders that are as rare as one and potentially without the need for large cohorts or trio data. But I would like to finish with a comment on where we are going next and what kind of things are we thinking about next. Certainly, severity is not one-dimensional, and what interpretable models mean can change very much. Certainly, there's 3 topics that are very important. I didn't talk about inheritance patterns, and the model that I've just described has some difficulty in distinguishing if a variant is mildly pathogenic but associated with dominant disorder or severely pathogenic in a disorder that is recessive. This is something that the model has some troubles dealing with it, and this is definitely an axis that we're interested in exploring. Variable penetrance is also another axis in this severity, non-one-dimensionality, and certainly something very interesting, and again, pointing to what Tim was talking about. There will be different mechanisms and molecular mechanisms driving disease and driving this pathogenicity, and it would be great to develop methods that can disentangle all of these different mechanisms. And all of these areas are areas that I believe really would benefit from models with explicit model assumptions, so that you can find more interpretable solutions for these predictions. And this is something that we are currently working on in our group. And this is CRG in Barcelona. This was work that was done in my group at CRG and also at the group of Deborah Marks at Harvard. In particular, Rose, she's a PhD student that worked very closely with this work. So thank you very much.
Hi, do we have any questions? Just in case there are any.
Yes. I don't know if you will be
There,
given
sorry.
a microphone. Yeah.
It's for the people online.
So one of the standard ways to create interpretable scores in machine learning is to calibrate the scores using techniques like isotonic regression. How is your method different?
Sorry, can you repeat your question? I'm not sure if I heard it.
So often people calibrate scores in order to make them interpretable using methods like isotonic regression. And so I was just wondering how different your method is from those.
So this method really just uses the presence of or not of a variant in the UK biobank in the human population. So really it's reminiscent to methods to assess the how the essentiality of genes, so methods like SHET or PIL-I, where you're just basically looking at the amount of loss of function variants across the population in a gene. These methods are very powerful, but they're really only gene level statements. And here we're just trying to use something that has the same sort of philosophy, but that will allow us to have variant level resolution. But that's kind of the calibration, quote unquote, is just really leveraging variation in the human population.